To come up with a viable mechanism for the conversion of alcohols to alkenes under strongly acidic conditions, we must consider the clues provided from various experiments. Firstly, we know that the reaction is reversible and therefore governed by thermodynamic ideas, with the alcohol being favored at equilibrium. The extra sigma bonds are stronger than the pi bond in the alkene. Next, if more alcohol is added, the reaction goes faster, while the amount of water present does not affect the rate, but does affect the equilibrium position. Importantly, as the alpha carbon becomes more substituted, for example in going from secondary to tertiary alcohols, the reaction rate increases. In terms of regioselectivity, the more substituted Zaitsev alkene usually predominates, and for stereochemistry, the trans isomer is favored when possible. Lastly, an important extra clue to the pathway comes from experiments in which the carbon skeleton is observed to rearrange. While this doesn't occur in all systems, it adds weight to the formulation of the mechanism. Now we are able to put the evidence together and work out what pathway is most likely to be followed. We know that the reaction is reversible. Indeed, alkenes are known to react with aqueous acids to give alcohols. This means that the reaction is controlled by the concepts of equilibria. Since the reaction goes faster if more substrate is added, but is unchanged if more water is present, the pathway has a unimolecular slow step. The rate also increases as the alpha carbon becomes more substituted, which agrees with a carbocation being formed in the rate-determining step. Carbocations are stabilized by electron-donating alkyl groups by hyperconjugation, as are the transition states leading to them. Zaitsev regioselectivity is likely the result of electron donation in the transition state of the deprotonation step, with more alkyl groups helping to make that route faster, which is based on electronic stabilization. While the preference for trans-stereochemistry is likely governed by the need to minimize steric interactions between substituents in the deprotonation step. An extra piece of important experimental evidence is the observation of skeletal rearrangement in certain substrates. This will be a very useful clue in determining the mechanism. Now we must put the clues together and come up with how it all matches one of the possible pathways. The highly acidic conditions are likely to boost the ability of the hydroxyl group to leave, so regardless of which direction the reaction goes, the first step will be a fast protonation to form an activated hydroxonium ion. After that, considering which bonds have to form or break, the water group is lost, a beta hydrogen is removed, and a pi bond is formed. This may occur through either a stepwise process, through an intermediate species, or a concerted route in which all bonds form and break at the same time. A stepwise pathway in an acidic environment suggests a carbocation intermediate, which could then be deprotonated by solvent to produce the alkene. Otherwise, a concerted process would involve beta deprotonation and loss of leaving group happening at the same time. The evidence points to a stepwise pathway with formation of a carbocation. This agrees with the observed rate data as well as the rearrangement possibilities. Having concluded that the mechanism is most likely to be stepwise, we need to come up with curved arrows that describe the bond breaking and bond forming events. We know that the powerful acid will protonate the hydroxyl first to produce the activated hydroxonium ion. The water-leaving group then breaks away, with the arrow showing the bond electrons going to the more electronegative oxygen and away from the less electronegative carbon. This produces the 6-electron carbocation in a logical way, which has flattened out into a trigonal planar shape and which is stabilized by hyperconjugation. If the carbocation were then to be trapped by water, the starting alkoxonium ion would be reformed in what is an SN1 nucleophilic attack. However, if a molecule of water acts as a base and deprotonates at the beta carbon, the pi bond of the alkene is formed. The arrows for that part of the mechanism begin with the water attacking the beta proton as a base and the C2H bond electrons being pushed towards the electron per carbocation. Those events result in each atom involved attaining an octet and thus stability. Notice that only one arrow is needed to represent breaking the C2H bond and forming the pi bond of the alkene. Since each of these events in the mechanism is reversible, the protonated alcohol SN1 product should predominate at equilibrium. However, the reaction is run at high temperature, so the alkene product may be removed from the reaction mixture by distillation. The equilibrium will then shift to compensate, according to Le Chatelier's principle, and more alkene will be formed. To summarize how the E1 mechanism is established, we run through how the data matches the accepted sequence of events. The fact that the rate-determining step is unimolecular means it must only involve the collapse of the oxonium ion with loss of the water-leaving group to give the carbocation. A carbocation intermediate would be then able to rearrange, which explains the skeletal reorganization observed in some systems. Such an intermediate would be able to go on to deliver both the regiochemical and stereochemical major products that usually result under these conditions. Also, formation of a carbocation is backed up by reaction rates increasing with more alkyl groups attached at the alpha carbon. This would stabilize both the intermediate and the transition state leading to it in the slow step. When we combine all of that data, the only pathway that makes sense is the unimolecular elimination known as E1.